The last talk of the day is Lizzie Wolkovich. Lizzie and I actually have worked together since grad school, so many years, and it's great to have her back in the neighborhood. She was in British Columbia and California and other places, but it's nice to have her back east again. And so she's going to be talking about her work on the race for spring and climate change in plant communities.
Can everybody hear me? Do I appear to be on mic? Great. Thank you, guys. It's great to be here. Thanks to Abe and everybody for organizing this. I have to warn you all, I have a tiny bit of a cold, so there could be a moment of coughing, but hopefully we'll get through it together. I want to open with this picture, which is actually, as you probably can figure out, me, well over 10 years ago. This is the first year of my PhD. I did my PhD out in Southern California, in San Diego, in one of the regional parks and one of the national wildlife refuges as well, so two different areas. But the reason I show you it is because I want you to see the landscape in the back. So this is coastal sage scrub, the habitat I worked in. This is a pretty average-looking year. It was a dry year, but most years are actually dry years if you think of how many are actually the dry ones. You can see there's a lot of scrub habitat and there's a lot of sort of dead brown stuff from the year before in between all the scrub and shrub habitat. This is the next year. So relatively the same time of year. This is the exact same park. It's just one of those hillsides you saw in the back before. And this is an above average rainfall year. By no means huge, but definitely above average. And then we had one year where we got the mean rainfall, right? Quite a rare thing to see in California. We had a huge wildflower bloom down in the valleys. And the reason I show this is because I think it really highlights the capacity that landscapes have for rapid year-to-year temporal change. We can see dramatically different things across landscapes. We think about this a lot in the West. We think about it a lot with drought-controlled systems. But I would argue in the research fields, we're finding more and more evidence that there really are critical climatic events that can cause rapid changes in landscapes, even in northeastern forests. So this is a picture from Harvard Forest, where I have projects that I'm just starting up now. And I would encourage you, if you're interested, to look at the work of Neil Peterson and other colleagues, really finding that climate shaping dramatic changes in a landscape is not solely the property that we see of drought-controlled systems, but is also a property perhaps of many, many systems as well, including northeastern forests. And so these short-term climate effects, I would argue, have been going on for over 10,000 years. But of course, what's happening now is that we've layered onto them a major trend with climate change. And I think it's finally and very fundamentally challenging ecologists like myself to answer, how does that short-term variation play out? How do we use it to understand long-term change? And how could those two factors be quite different predictions, basically? And so just to remind you all where we're at with climate change, this is a figure from the Intergovernmental Panel on Climate Change from the 2007 report, but the report that came out just last fall has very little differences in this figure. On the bottom it shows, the legend here is degrees Celsius in addition to the warming that we've already had on Earth. So we've already had about a 0.6 to 0.7 degrees Celsius warming event over the last 20 to 30 years. And what we're expecting to see by the end of the century is anywhere between a slight cooling all the way up to a 7.5 degree warming event in the Arctic and other areas. All I want you to take away, though, is just that most of the terrestrial landscape, we are predicting a 2 degrees Celsius warming event within the next 80 years, which I think is a dramatic change that we don't really have the best abilities to predict. And so I think scientists like myself, and I'm sure many of you in the room, are grappling with this very fundamental and pressing question of how do we and how can we predict responses with climate change. And so for my research, I focus on trying to do that by better understanding phenology. So I'm sure all of you know, but just to review, phenology is the timing of recurring life history events. So we think of the day a butterfly comes out of its cocoon, the day a bird starts its migration, or if you're me, I work primarily on plants, so I think about the timing of flowering, the timing of first leaf, and that's what I'll talk predominantly about today, the timing of spring events for the most part, especially in the beginning of the talk. And what I'm going to do is 3 quick vignettes. Well, we'll see how quick they are. 3 vignettes of different areas of research I've been working on over the last 5 years. I'm going to open by just talking about our best estimates of the direct abiotic response to climate change in the phenology of plants, their timing of leaf out and flowering. And then I'll talk a little bit about what might be evidence for very strong indirect effects actually playing out. So not just thinking about how did plants shift their phenology, but what are going to be more of the community consequences of that. And then at the end, I want to talk about non-stationary responses. And a simple way to think of the term non-stationary is An outcome if things are non-stationary is that we can't very well use the past to predict the future. We end up somewhere where we really have to understand the mechanisms to know what's going to happen moving forward and how dramatically landscapes, and in this case I'll use an agricultural system, may change. Okay, so first off, thinking about simple, direct, abiotic responses. I've always argued that this should be something that in the sciences we can tell you a very good answer for, because phenology is the most reported biological indicator of climate change. We have by far the most data on plant phenology to tell you how much it's shifted over time, and in particular with anthropogenic climate change. Part of the reason is that we have tremendous records. I'll talk more about this record in the background here in a little bit. This is wine harvest records starting back with the monks in France that goes back over 600 years. This is a record from New York, goes back over 100 years. These were just citizens going out to their backyard time and time again collecting phenological data. Marsham from England, who started a record that now goes for over 200 years. And then we also have a lot of sort of more scientifically initiated studies. So the International Phenological Gardens in Europe, you can see many, many sites started by academics in the 50s and 60s with a very rigorous methodological scientific approach. The Cloned Plants Project, for example, in the US. And we've used these data sets often in a more piecemeal fashion to really be the evidence that we have for the impacts of climate change on plants, with a lot of major papers coming out and finding in general species tend to shift their timing earlier and earlier as it gets warmer and warmer. But one thing I was really interested in when I started working in phenology was trying to bring together the data and look at how consistent it was. Do we have a relatively consistent answer for how plants have shifted their phenology with climate change? And so I built a database here with a bunch of colleagues, including Abe, who put in tremendous efforts. It's shown in blue. It's called the Nectar database. The size of the circles represents how many species are in that in each site. So this is the Henry David Thoreau and Abe's and Richard Premack's data set here in Concord, which has tons and tons of species. Thank you, guys. And a few others. Fargo has a good number in Washington, D.C. as well. This is a record out of England by Alistair and his father Fitter. And we combined all of these together, and they're all wild plant species, which is relatively novel. And we had over 5,000 species represented with time series data for about 1,500. Time series meaning multiple observations over 10 to 30 years. And then there's another data set here shown. This is the PEP data set out of Europe, so a major international effort to bring together hundreds of thousands of time series data. So there are hundreds of thousands of dots hidden here. Our data set didn't have that many. I think we have like 17 shown here, but it really was trying to pull together the majority of the data we had in Europe and North America and look at what we could see. And so what I'll show you is data on what I call temperature sensitivity, or the change in days per degree Celsius. So that metric is shown here on the vertical axis. It ranges from -10, so a species has advanced 10 days per degree Celsius if it's at -10. The 0 threshold means no change, and then a 5 would mean a species that has delayed, so it's actually flowering or leafing later, 5 days per degree Celsius. And the first thing I'll show you is observations from this relatively site poor, so only about 15 sites, but about 2,000 or so species. I think for this analysis, just about 1,500. The Nectar database, and I'm showing you means and standard errors. So we've pulled all the different data across all the sites and species. And what you see is that species have advanced their flowering or their leafing by about 4 to 6 days per degree Celsius, which was actually dramatically in line with the previous estimates coming out of Europe. So this is that European database that I just mentioned. You can see they have hundreds of thousands of time series. They have almost no error when you go out and calculate the mean in that case. But it is strikingly similar in my mind, given how different it was. There's only about 100 species in this data set. Lots of the data came from extremely rigorous academic methods for phenology. Lots of our data came from Henry David Thoreau, random people in New York, as well as more scientifically collected, if you will, data from researchers funded by a university who went out and did it versus people who did it for their own personal enjoyment. This was a study where we were also looking at experiments, so short-term experiments where people have gone out and actually warmed the landscape to try to see what they would predict from those studies as compared to long-term records. I'm not going to focus on this work, but I am most happy to tell you about it, and I'll just tell you the take-home here. The experiments are shown in red. The Nectar data is still here in blue. And what we found is these short-term experiments predict very, very little change. So on the scale of half a day to 1.6 days per degree Celsius. So if you were going to project what you expected to happen with climate change, you would get dramatically different answers, whether you used experiments or observations This graph on the left is showing all the species in the data, about 1,634. We narrowed it down to just the 36 species that occurred in both experiments as well as in long-term observational data. And you can see what happens here. Again, I just wanted to stress, these are incredibly consistent means. The errors go up when we reduce our observational number, but we still get about 4 to 6 days per degree Celsius. For the experiments, we get an even more drastic difference between experiments and observations. So one thing I hope you'll take away from this is that so far, combining hundreds of thousands of time series data over several thousand species, we have a very consistent mean estimate. I think we should be happy with how well we're doing on this. If you want to know how the average species is going to change, it's going to change 4 to 6 days per degree Celsius, especially in the temperate zone. But what's hidden behind this data is a lot of scatter. So this is a figure showing every single data point. So each species within each site goes into this figure. So you're seeing, as we look at the long-term data from the Thoreau data set, for example, what does that actually look like? No, try that. I don't know why the microphone keeps going off and on. Sorry, guys. But what I want you to see here is take a look at the vertical axis, which was just ranging between -10 and 5 days. And it's now gone from well over a month advance at the highest to almost a month delay. So there's huge diversity across species, and I would argue that we don't have very good abilities to predict it. One predictor is shown here. This is the mean flowering date for each species. So species coming out very early in the spring up here, species coming out very, very late in the summer and into the fall back here. And we see that the most sensitive species, the species that tend to advance the most per degree Celsius of warming, are the early species. But I think we really need to push ourselves here to better understand and predict this if we want to move forward and think about how do we actually take scientific data and move to conservation and mitigation with it. Because behind the average that looks great is a ton of scatter. Okay, and next I wanted to think about one reason that could drive that scatter, which is that phenology could be critical to indirect effects so that species interactions are actually playing a role here. And so for that I started looking at invasion biology. I'm just gonna see if I can't just try this one more time. Okay, sorry guys. So I think invasion biology is a great area to start to think about this question because it's a sort of fundamental area. If a species can get into a system, if it can establish, we know something about, there must be traits of that species or possibly traits of that system that have allowed a species to get in and establish. I'm sure you know a lot of these major invaders. Purple loosestrife shown up here on the left, woolly adelgid shown down here on the bottom. This is the species I actually worked on for my PhD, Bromus madritensis subspecies rubens, or red brome. And what I basically asked was, how can we use invasion biology theory and start thinking about phenology to ask whether there's any evidence that phenology really matters to how species interacts and that better predictions from phenology would come from thinking about species interaction. And so there were several major hypotheses we put forward, but I think the one that has found the most support is this one, which I'll walk you through now. And I should point out, Abe has done a lot of work on this as well. You'll see a picture from Concord at the end of this. And so it's a very simple concept. If you think of each species, sort of a simple two-species system, here's an exotic species in blue, here's a native species in red, and we're thinking about them flowering, for example. So here's their niche. A few individuals flower very early, most individuals flower around now, and then there's a tail. And then here's the growing season. I would argue that with climate change, what we've effectively done as humans is extended the growing season. We've pulled it open, we've made it longer. If one of these species is actually more plastic or more flexible for whatever reason, and is able to track the start of the season better, so if it's able to just change its leafing or flowering earlier and earlier as climate gets warmer and warmer, what you would very simply predict is that that exotic species would move forward in the season and now has really all of this space to itself. So it's no longer overlapping with the native species, it's no longer competing with it, and in fact, it may be getting early access and priority access to all the resources in that system that it wants because it's coming out and growing in advance of the native species. And so one of the first studies to show this was out of Concord, Massachusetts with phenology data. And what I wanted to do was really try to pull together a bunch of different data sets, as many as we could, and say, is this consistent across the landscape? So if you try to pull together as many different data sets as you can, you end up with 4, because you need a very particular type of data set with a lot of species, and actually a mix of exotic and native species. And I think we tend to have an issue in ecology where we measure all native species, or a landscape with lots and lots of exotic species, and we don't seem to measure as many of those systems in between. So what I'm showing here I call flowering time shift. And basically it's showing you how many days a species has shifted over some time period. So on the top, it's days per year. So if we have data that started in 1970 and ends in 2010, we're looking at how much that species has shifted over 40 years each year. So if it shifts about a quarter every year, in 10 years, it's shifted 2.5 days total earlier. Just like before, negative means that species are advancing with warming, and over time, excuse me, which also coincides with warming, or species could be delaying if they have a positive value. And again, the 0 mark shows where there would be no change or really no effect. And the exotics again are in red and the natives are in blue, and I'll show you 4 different data sets. First one coming out of Washington, D.C. Again, these are means and errors for the entire plant community. So we take about 300 species, dump them in, assign them if they're native or exotic, and look at their differences. And what you can see is that here, indeed, the exotics have shifted more over the recent time period than the native species, not very significantly so. But if we start looking at more data sets, we'll see this is rather consistent in temperate mesic spots. This is Chenor, England. Here, the exotics have moved significantly earlier than the natives. This is the Concord, Massachusetts data I just talked about. Very dramatic change. Same concept here, but in this time it's total days over some time period. And so here for Concord, Massachusetts, it's about almost a week over the last 150 years versus native species, which really haven't shifted at all. We actually see the reverse in systems that are more controlled by precipitation. So for Fargo, North Dakota, we find that exotic species have actually advanced, while native species, again, haven't changed. And one thing that I found rather dramatic when I started looking at these data is that Chenor, England is a system with an abundance of exotics. It's actually quite hard to classify exotics in England. Whether or not all of these things are exotic is a debate you can have with anyone from England, because they have had species obviously coming in for so long. But if we look at the 3 North American systems, which we see is that native species have actually not shifted. So we find that across all these systems, the native community actually overlaps with the 0 mark, suggesting to me that the dominant change that we're seeing in phenology, possibly at least in North America, is driven more by invasive and exotic species than possibly by the native community. And I think that really calls on us to think about what the capacity for the native community is for change as compared to the exotic community. Okay, so hopefully that would suggest to you that I think there is room to start thinking about trying to better understand some of these differences by thinking about how species interact and how phenology is critical to that interaction. I want to close with some work that is hot off the presses, thinking about whether we see non-stationary responses, so whether we can really use the past to help predict and understand the future. And for that, I'm going to talk about wine grapes, actually, which is a new system that I'm developing, not because I want to work in wine grapes, which is a rough life, but because it's actually a tremendous model system for phenology. Wine grapes have more phenological variation than any other plant species I've found on Earth. They vary 3 months in their leaf out time and 2 months in their flowering time, and they are actually just a subspecies. When you drink wine, it is not even a true species. It's a very tiny amount of genetic variation that produces all the wines you see and produces huge phenological variation. The other reason to work in wine grapes is that they have some of the very best and very longest phenological records. So this is called the Daux wine harvest date record. It's a composite of a bunch of different records put together, and it just shows you over time. I'm just showing you a running average of an anomaly. So here's the mean, and then days, years of the harvest that were earlier versus later. There's a 10-year running average in red here. And again, I hope you're noticing systems are dramatically changing year to year. This is one of the challenges of communicating climate change. I think it's shown very well here. Here's the variation in the climate system. We know it's there, it's huge, it's tremendous. This is climate change. It is exceptional every time we check, but I think we also have to find a way to better communicate that there is a reality to how variable climate systems are. Again, not just in drought systems, but as well in temperate and mesic systems. So I'll very quickly go through questions we're looking at with this new data set. First off, very quickly, I'll talk about whether or not phenology has shifted in the last 20 to 30 years, which would be in step with climate change. How sensitive that shift is, and then talk a little bit about, is there evidence that we need to think carefully by using the past to predict the future. So just a little bit more about the record. It's 380 different sites. So 380 sites scattered all over France. These are harvest dates, so when the wine and the grapes are coming in for harvest. They cover several different regions that have different varieties of wine grapes, but a lot of spatial data as well as temporal data in this case. And the first thing I'll show you is the sensitivity. So this is gridded spatial data We use gridded climate data for those of you interested. I'm happy to talk more. But you can just take a look at this axis going from -1. This is standardized from 1 to -1. So things that are in red are delays. So when there is more precipitation across Europe, we tend to see a moderate delay in wine harvest. Things in blue are advances. So you'll see the temperature graph over here on the left is in blue. And what we tend to find is when it's hot in Europe, and these are for the summer months, which is the most predictive months for wine grape harvest, we tend to see an advance in the harvest. So very simply and straightforward what we would predict. Earlier, warmer temperatures, earlier harvest, more rain, later harvest. And just to show you quickly, this is comparing 1600 to 1979. Most reports of changes in the actual climate system occur between 1976 and 1984. So we're bracketing around 1980. The data before 1979 onward is here. And then here we're looking at the data 1980 to 2007. And the take home between these two histograms is really just here. On average, if you look before climate change really started, the average harvest was about 2 days more than the anomaly. Now it's about 10 days more. So a really dramatic change in harvests in France. But now I want to think about that non-stationarity. And what I'm going to do for that is I'm going to take the years where harvest was very early. So years where harvest was very early also tend to be years of a very good harvest. So we were interested in these years for that reason, and also because we wanted to understand what actually allows that. And they are very anomalous years. Over our long record, we only see 45 years where the record is actually about 10 days earlier. So we took out those years. There are 45 of them, as I said. And then what we did was we looked at the climate of those years. So we said, of these 45 years that we have that are very, very early, what does the climate actually look like? So this is the total look at that. Temperature is here again on the left. And as you can see, in this case, the negatives still are going to be delays. Delays, and the positive dark reds are going to be advances. So when it's warmer, we get an earlier harvest. Here I'm showing you something called the Palmer Drought Severity Index. You can think of this just as soil moisture. And so what we see here is that when there's a drought, we also tend to get earlier harvest. Very similar to what we saw with the basic sensitivity. What's dramatic is what happens when we compare those two. So I'll do it first with temperature. So here I'm looking at the 33 years that were before 1950, and here the 12 years that were after. So again, we're just thinking about these anomalously early harvest years, and comparing and contrasting those before 1950 with those after 1950. What did the climate look like in those years? And what you see is it's actually quite similar. Those were very hot years to lead to very early harvests. And you can see before 1950 they were fairly hot, after 1950 a little bit more dramatically hot. But the more interesting piece comes out when you look at the drought. So this is a contrast of that drought index that we can more simplistically think of as just soil moisture. What you can see is that before 1950, when you had a very early harvest, which again was often a very good, high-quality vintage, you had a major drought system in Europe in those years. Since 1950, there's really no correlation with drought when we get an early harvest. So what's fundamentally happened in this system, as far as we can tell, and again, this is new, we're still working on it, is that before, all the way up until sometime after 1950, if you wanted a really warm summer, it also included a drought, and that's what led to an early harvest. So warm summers came with drought. That's fundamentally how the climate system appears to have worked. Since then, we can get extremely hot summers without a drought because we have such an increase in the temperatures in the climate system. And I would argue that it sort of fundamentally shifted what controls wine grape harvests in Europe at least, and we're trying to expand this to other systems. And I'll close very quickly with one thing that I think a lot about lately. Going back to my PhD system, this again is San Diego. This is an aerial view of San Diego looking down on the interstates as well as the urban systems. If you can see, there are little fingers of habitat within all of this urbanization. These are canyons that are too steep to build on. And when I started my PhD, I was fundamentally interested in this process of fragmentation. What happens to organisms when we fragment the landscape? How can we understand and predict the effects of urbanization, degradation, and habitat loss? And when I went into my schooling and started to learn about this, there were a ton of theories to draw on in ecology to think about the outcomes of habitat change. Island biogeography, metapopulation, metacommunity, onward and onward. I would argue that climate change has fundamentally changed the temporal space that organisms have. We have radically changed the growing seasons, and we are only going to more radically change them in the future. And I think ecological theory has very little to offer in how we understand and predict that, and it's an immediate area that I would argue people need to start thinking about. We made headway in thinking about what are the spatial consequences. I think we need to make a lot more headway in thinking about the temporal consequences. I'll stop there. Thank you, guys.
Thanks. So we have time for maybe one question before we get into the panel discussion thing.
Awesome talk. The lack of short-term change. So the change in how change is happening.
Yeah.
Is there any evidence that there's either a phylogenetic signal or that there's genetic changes going on in these populations that are changing the way they respond to climate?
I have two pieces of information. The phylogenetic work I think is really hard. So Abe and I have both been involved in some of that work, and what you're doing with the phylogenetics is you're taking usually the day of year that something leafs out and putting it on an evolutionary tree, when you know that that day of year varies across the species range. So I think the only way to make headway and know how much phylogenetic signal there is to get down to the cues and say, this is the photoperiod cue, this is the temperature cue, and look. There already is signal, so I think there is a little bit of predictive power for sure in how related species are, and I think we need to do a better job of understanding and using that. The other thing in terms of actual genetic change is people are just starting to work on it, but my suspicion is there is a dramatic amount of genetic change probably happening in the invasive populations. There was a new paper at the beginning of the year on purple loosestrife that doesn't have conclusive evidence because they obviously don't have seeds from when the species came in at the very beginning, but has very strong evidence that that species has been successful as an invader because of its phenology and fundamentally because it's able to adapt and genetically change its phenology very, very rapidly.
Great. Thanks so much, Lizzie. Really appreciate it.